Good morning. Good morning, dear participants of our conference. We start our third day, and this day is dedicated fully to the hepatitis E. And at the opening ceremony of the conference, we told that it will be dedicated to the 40th anniversary of the experience of this case, which was done by Mikhail Balаяn, members of this conference. I think that because of this, our conference is the best one, and any conference which is dedicated to the virus hepatitis E will be envy on the reports which will be done by the outstanding people who made very sufficient support. Let me also tell a good morning, a good time of the day, dear colleagues. Someone has a morning, someone has a day, and we are glad that you today are with us. And of course, Mikhail is absolutely right that today is different to other days, because this day is very, very interesting and rich in anniversary dates. Yes, sorry to interrupt. Yes, thank you. According to our tradition, once in 2 years at our conference, we give the award, the medal named after Balаяn, Mikhail, and we should say that this person was really outstanding one. And as one of the specialists of this hepatitis E study, he said that Mr. Balаяn was the brilliant of the world science, who made very important development of the study of hepatitis E and his self-infection. That was tremendous stage of the study, and that made a push forward to many other events and things. In the next slide, please. Several years ago, on the 80-year anniversary, the international group of the scientists decided to actually start the international gold medal and to give this medal for 2 specialists from the Russian Federation and for 2 foreign specialists. Sometimes we give even more medals, but this medal is given once in 2 years. And I should say a couple of words directly about this medal. This medal is done by the great outstanding artist, one of the best specialists in making the medals, Alexander Kolotkin, who made this medal, and his medals are in museums of Hermitage, in Louvre, Metropolitan Museum in United States of America, and also in museums of Russia. This is the world famous person who made a lot of beautiful medals. And this medal also became one more piece of art which we are glad to award to the winner of this medal named after Balаяn. When this was organized, the international committee decided to give the first medal to give this medal to the family of Mikhail Balаяn, to his daughter, and his family has this medal. And after that, they were announced the 2 winners. The first 2 winners in 2012, there was a great specialist, Professor Mohammed Horu, the Indian researcher, the first one who described virus hepatitis E in Kashmir outbreak. And today you will enjoy his lecture at our session. And the second person who was given this medal was our specialist, our big friend Alexander Anjaparidze, who together with Mikhail Balаяn made the study of this virus. And you will listen to his report today also. Then there were awards from CDC. There was 2013. The medals were received by Salim Kamili. His lecture, his speech you will listen to today. It's about laboratory diagnostics. Mikhail Favorov, he also was given an award for the study of the outbreak in Uzbekistan and Tashaus. Also Jean Drobenik, who made great support in CDC in the study of virus hepatitis. His first publication was on the reveal of the high level of antibodies on the virus hepatitis E or the workers who have the contact with animals. Dr. Yuri Karyatni, who made a great study on the molecular diversity and genetic diversity of these people who were working on hepatitis E in the group of Russian specialists. This is Dr. Ogarkov for the studies of hepatitis E in Afghanistan, Tatiana Bystrova for the study of hepatitis E in Russia. She's the student of Mikhail Balayan. Also, Mr. Pokrovsky, who studied hepatitis E, and Gennady Onishenko, who had PhD dedicated to the topic of hepatitis E, and also your reverend speech maker now, Mr. Mikhailov. In 2016, Dr. Mamedov received the medal for the study of hepatitis E in group of Balayan. In 2018, there was a new group of the specialists who received this. This was Dr. Zhavarnuk for the study in Belarus, this fundamental study on hepatitis E. We are going to listen to his speech. Then Yelena Malinikova for the cycle of the works on the study of the clinics in hepatitis E, on the Heparin in study on the first not endemic outbreak in Russia, in Kovrov city. Stanley Lehman for the cycle of works and for the opening of the new form of the virus of hepatitis E. Helena Norder, our great friend, who is making this study in Europe and Sweden. There is a cycle of works dedicated to hepatitis E from her side. In 2019, there were also medals given to Dr. Kurigyan for the cycle of works on the hepatitis E, and for two specialists in research, Dr. Burkov in establishment of ecosystem and industrial production of these test systems, which are and Dr. Abryadina who made the diagnostic agents and tools for the study and diagnostics of hepatitis E. Also, the medal was given to Daniel Bradley for the study of hepatitis E and characteristics, and our big friend, the person who made the study of different virus hepatitis and hepatitis E especially, Michael Porty, professor, for the original study of hepatitis E. These are the winners which were given the medals. All these medals are unique, and everyone get the diploma and the medal with his own number. This year, as the nominees, they were introduced 4 people. This is Dr. Meng from United States of America for the first work on the study and reveal of the hepatitis E in animals, and he was a leader in this question. His classic works are the part of International Foundation of the works dedicated to hepatitis E. Also, the medal. Dr. Meng will make a speech today, by the way. Dr. Jamil from India. He made the cycle of work on the molecular and biology study of virus of hepatitis E. His fundamental knowledge enlarged our picture of the world dedicated to hepatitis E, and our 2 Russian specialists. This is Yuri Karyatni, who worked with Mikhail Balayan, and he also made significant support in the study of hepatitis E and the animals. He is receiving medal, and also Ilya Gordichuk, Dr. Gordichuk, in the Institute of Polymyelitis for the preservation of the colony of the monkeys, which was established by Mikhail Balayan in the cycle of works for the experimental modeling, and he is going to tell us today about the establishment in the institution of the polymyelitis, the vaccine against hepatitis E. So, And now we move directly to the awarding ceremony, and we also going to listen to the speeches of person who were awarded. The first one, who are we going to give the medal. This medal will be given. This medal will be given to the person who is receiving this award. This diploma is also given by their international committee, and the first one who is given this medal, this is Dr. Meng. It was a great pleasure. I turn the floor to him for the speech about his study of hepatitis E.
Hello, everyone. It is a great honor and a privilege to receive the 2021 Balayana Medal. So I would like to thank members of the selection committee for this honor. Dr. Balayana was truly an inspiration for many of us in the field of viral hepatitis. His self-sacrifice leading to the discovery of HEV will be forever remembered by future generations of scientists. I have been personally studying different aspects of HEV for more than 2 decades. But today I would like to very briefly discuss our very early work in discovering the first animal strain of HEV and the impact of that initial discovery on the field, which, as you know, eventually led to the recognition of hepatitis C as a zoonotic disease. Now, prior to 1997, the year in which we discovered the first animal strain of HEV, a number of seroepidemiological studies had reported the detection of HEV antibodies in blood donors from many industrialized countries where hepatitis C was not known to be endemic. As you can see from these slides, HEV antibodies were detected from blood donors in a large number of industrialized countries, ranging from 2% in Australia in 1995, 2% in the Netherlands in 1993, to approximately 18% in the United States in 1997. So these findings were very puzzling at the time, since HEV was not known to be endemic in those industrialized countries. So the question was, where do these infections come from? It had been speculated at the time that an animal reservoir may exist, which then would explain the seropositivity in blood donors from non-endemic countries. So our group discovered the first animal strain of HEV in 1997 from pigs in the United States, and we designated the virus swine hepatitis C virus, or swine HEV, to distinguish it from the human HEV. Now, the discovery of swine HEV actually is very serendipitous, since at the time I was not even working on HEV at all, and I was a scientist at the National Institute of Health in Bethesda, Maryland, working on the hepatitis C virus at that time. So in early 1996, Dr. Dale Webb, who is a veterinary pathologist from the Illinois Department of Agriculture, contacted me about some hepatitis pathological lesions he saw in the liver of some pigs. So I asked Dr. Webb to send me some pig serum samples, and we then tested them for antibodies against a panel of known hepatitis viruses. And to my very surprise, many of the pig serum samples we tested were found positive for antibodies to the HEVs. And I recall that this finding was very confusing, very puzzling at the time. And I remember I had multiple discussions with Dr. Robert Bissell and Dr. C. Emerson, who were the viral hepatitis lab chief and the section chief at the time at the AIH. Why would the pig be positive for antibody to HEV, which was not known to be endemic in humans in the United States, let alone in pigs? So in order to solve this puzzle, we conducted a prospective study in a pig farm in the state of Illinois, in which we followed 20 piglets from the time of birth until 6 months of age. And surprisingly, we found that the majority of the pigs older than 3 months of age were positive for HEV antibodies. Pigs naturally infected by the swine HEV had microscopic lesion of hepatitis. They developed viremia prior to seroconverting to HEV antibodies. And importantly, when we sequenced the virus, we found that its capsid gene share about 80% nucleotide sequence identity and 90% amino acid sequence identity with the human hepatitis C virus strains. Now, showing here in this slide are 3 representative pigs from that initial prospective study. One pig was born to a seropositive cell with a high level of HEV antibody, and one pig was born to a seropositive cell with a low level of HEV antibody, and one pig was born to the seronegative cells. And as you can see here, at 2 weeks of age, the pig born to the seropositive cells with a high level antibody was seropositives at birth, and then the HEV antibody gradually decreased and went at about 7 weeks of age and become undetectable between 7 to 14 weeks of age. So clearly, the antibody we detected in this pig at the birth and also at the beginning of the pig's life from 0 to 7 weeks of age, these are maternal antibody acquired from the seropositive mothers. Now, if you look at their at the 15 weeks of age, which is about 4 months old, this particular pig become naturally infected, seroconverted to HEV antibody, and remain positive throughout the remaining period of study. Now, the other 2 piglets you can see here, they were born to either the seronegative cell or seropositive cell with a very low level of maternal antibody, and so they are seronegative at birth, but both also become naturally infected and seroconverted to HEV antibody at about 18 weeks of age. So this prospective study clearly demonstrated that swine HEV is naturally circulating in the pig population, and most of the pigs become infected by the swine HEV at about 2 to 4 weeks of age. Now, the infected pig appear to be clinically normal with no overt clinical disease of hepatitis, but they do develop microscopic lesion of hepatitis. And you can see here from this slide, the liver of the infected pigs had multifocal lymphoplasmacytic and necrotizing hepatitis. And they also have lymphoplasmacytic inflammation, as you can see here in the lower left corner of the slides. And you can see some of the sweating and evacuation of the hepatocyte, and also you can see the foci of hepatocellular necrosis. So they do have microscopic lesion of hepatitis, even though the infected animal are clinically normal. Then subsequently, in 1998, we reported the experimental evidence of cross-species infection for the first time by the swine HEV. And you can see here from this particular study, we demonstrated that swine HEV from pigs can readily infect non-human primates, both rhesus macaques and chimpanzees, which are the surrogate of a human. So this critical virus transmission study indicated that hepatitis E is likely a zoonotic disease, and the pig is animal reservoirs. So shown here in this slide is a rhesus monkey experimentally inoculated with the swine HEV. As you can see, this rhesus monkey clearly become infected as evidenced by seroconverting to HEV antibody at about 4 weeks post-inoculation. And also we see the viremia and the fecal virus sheddings starting at 2 weeks and lasting about 5 weeks. And there's also the elevation of liver enzymes, both ICD and ALT at about 4 to 5 weeks post-inoculation. And during this peak liver enzyme elevation, you can see here, we also observed hepatic pathological lesion in the liver, which is consistent with viral hepatitis. So clearly, this non-human primate become infected by the genotype 3 swine HEV. Now, conversely, we also show that the genotype 3 and the genotype 4 human HEV also infect pigs. And as you can see here from this slide, pig experimentally infected with the genotype 3 human HEV, isolated from the patient in Minnesota, and with the genotype 4 strain of human HEV, isolated from the patient in Taiwan, quickly become infected. They all seroconverted to HEV antibody at about 2 to 3 weeks post-inoculations. And the infected pig also become viremic and shed virus in the feces. So the result further provided further evidence that hepatitis C is a zoonotic disease and the pig is the reservoir. Now, swine HEV infection is highly prevalent in the pig population worldwide. And about 80% of the pigs in some herds here in the United States are infected by the swine HEV. So this really raises important concern for zoonotic HEV infection through direct contact with infected pigs or through consumption of undercooked contaminated animal meat. So shown here in this study is a very recent work from my group showing that pork supply chain at the entry point, which is the slaughterhouse, is contaminated by the swine HEVs. And in this study, we tested more than 5,000 market weight pigs from 25 different slaughterhouses in the United States. And if you just look at the bottom line summary of these slides, you can see here that more than 6% of the slaughterhouse pigs are still viremic. There are still virus circulating in the blood at the time of slaughter. So this suggests that HEV containing blood at the time of slaughter may contaminate the pork products, leading to potential foodborne zoonotic infections. And in fact, large number of cluster or sporadic cases of hepatitis C have been definitively linked to the consumption of undercooked animal meat, particularly pork products. Showing here is one example of the cluster cases of hepatitis E acquired through the consumption of a pork liver sausage and fugitello, which is a popular sausage in southern France. And you can see here 3 family, they get together and had a party. And the pig liver sausage fugitello was served at the gathering. Of the 13 family members who ate the fugitello sausage, 7 of them become infected by the HEV. However, none of the 5 family members who did not eat the sausage had any evidence of infection. But importantly, the HEV sequence recovered from the leftover fugitello sausage had essentially identical sequences to the human hepatitis C patient. So this indicates that the source of HEV infection in humans came from the fugitello sausage. And in fact, foodborne HEV infection has been reportedly in many different countries worldwide, making newspaper headline like the one seen right here. This is from United Kingdom. Said UK supermarket may have infected thousands with hepatitis C from sausage and hams. Now, in addition to swine HEV, our group also identified the avian hepatitis C virus or avian HEV from chickens in the United States. In 1999, Chris Payne from Australia first reported the amplification of very short fragment of HEV-related sequence. It's about 500 base pair, I believe, and from chickens with a big liver and a spleen disease. So 2 years later, my lab was able to first visualize 30 to 35 nanometer HEV-like particles. You can see here under the electron microscopy. And the virus particle we see is from the bile of chicken with hepatitis splenomegaly syndrome disease in the United States. And importantly, we successfully determined the complete genomic sequence of the virus, which led to the discovery of avian HEV from chickens in the United States. We found that avian HEV infection in chickens in the United States is widespread, and avian HEV share only about 50% sequence identity with the human hepatitis C virus. Importantly, we were able to experimentally reproduce hepatitis splenomegaly syndrome disease in layer chicken experimentally infected with avian HEV. And you can see here from this slide, the gross liver lesion on the left in some of the infected chickens are characterized by subcapsular hemorrhage or by the enlargement of the liver. And so it's the characteristic disease for the hepatitis splenomegaly syndromes. And the microscopic liver lesions was mainly lymphocytic paraphlebitis and phlebitis. You can see here from the right side of the two micrographs. And we further demonstrated actually that even HEV can cross species barrier and infect turkeys, but it does not infect rhesus macaque. So this suggests that even HEV is likely not zoonotic and probably not infectious in humans. But regardless, the discovery of even HEV and the reproduction of the hepatitis splenomegaly syndromes in infected chicken provided the scientific community with a very unique and useful animal model system to study the clinical aspect of hepatitis E, because even HEV infection in chicken is associated with a clinical hepatic disease. Now, since our initial discovery of swan HEV from pigs in 1997, novel strains of HEV have been genetically identified now from more than a dozen other animal species. Showing here in these slides are some of those well-known animal species from which HEV has been genetically identified. You can see here the domestic wild pigs. They have genotype 3, genotype 4, and also genotype 5, genotype 6 HEV strains. Camel, which harbor the genotype 7 and genotype 8 HEVs. The deer and chicken, which we mentioned earlier. There are now at least 4 distinct genotypes of HEV in chickens, rabbit, rat, and even fish. And so it is likely that additional novel strain of HEV will continue to be discovered in the future from other animal species. So the discovery of a large number of animal strains of HEV have greatly expanded the host range of the virus and led to reclassification of HEV in a new family called Hepiviridae. And as you can see here from this slide, the left panel, and the Hepiviridae has 2 distinct genera. The genus Orthohepiovirus, which consists of 4 species, Orthohepiovirus A, B, C, and D, and genus Pisahepiovirus, which is represented by a single virus from fish called cut-throat virus. Now, HEV strain in the species of Orthohepiovirus A and Orthohepiovirus C are known to infect humans. So if you look at the slide on the right panel, which show 8 different HEV genotypes within the species Orthohepiovirus A, and you can see here that the genotype 1 and genotype 2 HEV infect only humans, and genotype 3 HEV infect human and a large number of other animal species, including deer, pigs, mongoose, and rabbit. Genotype 4 HEV infect human and pigs. Genotype 5 and 6 infect wild pigs. And the genotype 7 and 8, as I mentioned earlier, infect camels and likely human as well. So HEV infection is highly prevalent now worldwide. And this slide shows the genetic different genotype of HEV and the 8 different genotype actually within the species also have virus A, and the geographic distribution of these viruses. And globally, you can see here, according to the WHO, hepatitis C remains an important public health disease. Each year, there are 20 million HEV infections worldwide, leading to about 3 million cases of acute hepatitis C and 44,000 hepatitis C related deaths. So this is still a very important public health disease. Now, in addition to the acute viral hepatitis, recently HIV-associated chronic viral hepatitis has become a significant clinical problem, especially in immunocompromised individuals, such as organ transplant recipient, HIV/AIDS patient, or patient with lymphoma and leukemias. For examples, approximately 60 to 80% of HIV-infected organ transplant recipients have been reported to progress into chronic hepatitis C. And most importantly, the chronic HEV infection are caused almost exclusively by the zoonotic genotype 3 and the genotype 4 HEV strain from animals. So this indicates that the chronic hepatitis C is of zoonotic origin, likely through the consumption of undercooked animal meat. Now, more recently, HIV infection has also been linked to extrahepatic manifestation of disease, such as neurological disease and the kidney disease. For example, approximately 5% of HIV infection have neurological disease, such as Guillain-Barré syndromes, neurological amyloatrophies. Similarly, extrahepatic disease are also linked mainly to the zoonotic genotype 3 and zoonotic genotype 4 HEVs from animals. So clearly, zoonotic HIV infection is now associated with important clinical problems. So in summary, since our initial discovery of the first animal strain of HIV from pigs in 1997, the hepatitis C field have evolved tremendously. And the textbook for hepatitis C has definitely changed, since we now recognize hepatitis C as an important zoonotic disease. And you can see here from these slides that more than a dozen animal species are now known to harbor the hepatitis C virus. And many of these animal HEV are also known to infect humans, causing zoonotic disease. However, the biology, the pathogenesis for many of these animal HEV remain largely unknown. So we still have a lot of work to do in order to fully understand the ecology and the natural history of this important human disease. So I'm going to stop here by acknowledging the people in the lab who did the works, and I have a large team working on this particular disease. And in the interest of time, I'm not going to name each one of them, but they are the one who did the work. And also, I have a large number of collaborators from different institutions. And also, our grant is primarily supported by the National Institute of Health and USD NAFOS. And thank you
very much, Dr. Ming, for this very interesting report about how this was studied. The only thing which I would like to add is that, unfortunately, when there was translation, sometimes we told hepatitis C or hepatitis E. The whole report is dedicated to hepatitis E, and I think that it would be interesting for all of us to see the person who made a big support to the study of hepatitis E, and we move to the next nominee, to the next person who received the award of diploma and medal named after Balаяna. This is Dr. Shahid Jamil, and this is his diploma and medal, which would be given to him from our side, and we definitely send it via post, all the awards. And that's a great pleasure for me to introduce his report about the molecular study of virus of hepatitis E. Please.
Hello and good afternoon, good morning, good evening, wherever you are. I'm very pleased to be able to talk to you about my work on hepatitis E virus. And I'm honored to be given the MS Balаяna Medal for hepatitis E research. I have followed Professor Balayana's work during my own studies and have been hugely impressed by his dedication to his field of research. My lecture today will be about molecular studies on the hepatitis E virus. The work that I'm going to tell you about today was carried out between 1988 to 2013 at the International Center for Genetic Engineering and Biotechnology, or ICGEB, in New Delhi. My work during these 25 years was funded by core funds from my institution, from an International Senior Fellowship of the Wellcome Trust UK, R01 grant from the National Institutes of Health, USA, and the Department of Biotechnology, Government of India. I would also like to acknowledge many PhD students and postdoctoral fellows and research assistants and trainees who went through my lab in these 25 years, as also numerous collaborators, some of the work which I will describe today.
Yeah.
So viral hepatitis, you are all familiar with. I just put this up for my own orientation to tell you that when I started my work, hepatitis E virus was not known as a virus. The disease was called non-A, non-B hepatitis, and the virus was known as enterically transmitted non-A, non-B hepatitis virus. Later, as you know, it became known as the hepatitis E virus, as opposed to the post-transfusion non-A, non-B hepatitis virus, which turned out to be what we call now the hepatitis C virus. My work really started with two separate studies, but before that, let me tell you about some of the research highlights. When I started at ICGB, it was a brand new institution. So we set up research facilities and a new research program at this institution. A lot of my time went on to set up facilities as well as to build both the research, lab research, as well as clinical research networks. During my studies, initial studies, along with collaborators, I covered 3 different outbreaks of enteric hepatitis, and these are shown on the map of India here. The first one where we analyzed samples was an outbreak in Kashmir, which I did with Professor M.S. Khuru and his colleagues. The second one was in a place called Yamunanagar in Haryana, not too far from Delhi, which I did with Professor J.B. Dilavri and his colleagues. And the third one in the southern part of the country in Hyderabad, which I did with Professor Habibullah at the Osmania General Hospital and Professor S.K. Panda at the All India Institute of Medical Sciences in Delhi. During these 25 years, I did several things, starting with transmission of fecal material into rhesus monkeys. We developed a PCR assay for hepatitis E virus, and that became very useful to do some other clinical research, which I'll tell you about. There were several clinical studies that my colleagues and I did, which are listed here. But then we also quickly got into cloning the viral genome and produced 2 different infectious clones of the virus, one from the Hyderabad outbreak and one from the Yamunanagar outbreak. A lot of time in my lab was spent expressing and characterizing the viral proteins. We did many functional studies with the open reading frame 3 protein, which is a regulatory protein in the virus. My collaborators and I did several studies to look at human immune responses to HIV infection. And towards the later part of my career, we focused on looking at host factors by carrying out proteomic and metabolomic studies, and finally to look at a potential receptor for hepatitis E virus and how the virus enters cells. I was also part of the International Committee for Taxonomy of Viruses, both as a member and as the chair of the HepiVirus group. And during that time, we worked on the phylogeny of HIV, as you know today. Okay, so let us get back to the Kashmir outbreak with Dr. M.S. Khuru and his student Salim Kamili. Both, I'm very pleased to see, are Balаяna medal winners in the past years. We did together. Dr. Khuru was really the person who first identified enteric non-A, non-B hepatitis as a disease by covering this outbreak in November 1978 in Kashmir. when we had set up the PCR assay and were doing antibody assays in the lab, Professor Kuru's PhD student at that time, Salim Kamili, came to my lab and carried out several analyses of those samples which they had collected during the Kashmir outbreak. And there were two papers that I list here that resulted from that, where we showed both long-term antibody status in hepatitis E patients, as well as a seminal study where we showed that hepatitis E virus can also be transmitted from a pregnant woman to her baby. The second clinical collaboration that I'll talk about is the one that we did with Dr. Dilavri and his student Ashok Chauhan, where we investigated the outbreak in Yamunanagar. Dr. Dilavri and Ashok were both at the Postgraduate Institute for Medical Education and Research in Chandigarh. Ashok was a very daring young man, and he followed in Dr. Balayana's footsteps. When he could not convince people that the material he had collected was from a hepatitis outbreak, he mixed the sample with orange juice and drank it. But then he also kept very good track of getting his blood sample and his fecal sample collected every single day, both before drinking the sample as well as after. And based on that study, when by the time we had developed a PCR test, we published this 1993 paper in Lancet, which was the which told us the natural history of hepatitis E virus during voluntary transmission in a human being. And later on, many years later, we also produced a full-length genomic clone from material collected from Ashok at that time. So based on these studies, we proposed a natural history of how the disease develops. And what we showed that was, even before symptoms appear, even before ALT rise, HEV RNA was found both in serum as well as in stool samples. The serum viremia was short-lived, whereas the virus continued to pass in stool even after the ALT peak had been obtained. By carrying out antibody studies, we showed that while the IgM anti-HEV rise was transient, the IgG was more long-lasting during the convalescence phase. But all this is now common textbook knowledge. Let me get on to molecular studies. As you know, hepatitis E virus belongs to the family Hepiviridae and genus Hepivirus. It's a 7.2 kilobase positive strand RNA, which is polyadenylated and capped at the 5' end. On the right, I show you electron micrographs from the stool samples that we had collected from outbreaks, and it turns out to be, an icosahedral capsid, like many other circular viruses. I also show you the phylogeny of this virus that we developed as part of the ICTV, and it has subsequently been revised in new editions of the ICTV reports. The virus, as we know, has both animal viruses as well as human viruses. And several animals are now found to be infected by this virus. Some of these seminal studies on animals were carried out by Professor XJ Meng, who I'm also very pleased to see is another awardee for the Balayana medal. So the virus has 3 open reading frames. ORF1, which codes for the non-structural proteins, ORF2, which makes the capsid, and ORF3, which is made from an overlapping open reading frame with ORF2, produces what we showed through multiple studies in my lab to be a regulatory protein that controls the host cell environment for viral replication. I'm summarizing here the life cycle of hepatitis E virus based on many of our studies as well as studies from other labs. The virus first binds to the surface of cells through attachment factors. And Manjula Kalia in my lab characterized at least the major attachment factor through which the virus binds to cells, and that is called heparin sulfate glycoproteins. These are heparin sulfate molecules attached to proteins that capture the virus through low affinity but high density interactions. Subsequent to that, the virus binds to a specific cellular receptor, and I'll show you some of the studies that we did in the later part of our work to characterize at least one potential receptor. Following receptor binding, there is receptor-mediated endocytosis through which the virus enters cells. Once it enters cells, it uncoats, the positive strand RNA is revealed, and being positive strand, it is first translated into the non-structural proteins. These non-structural proteins also include a polymerase, a RNA-dependent RNA polymerase, which converts the positive strand into negative strand shown at step 6. And these negative strands are then converted back to positive strands, either subgenomic positive strands shown at 7 or full length genomic plus strands shown at step 8. The subgenomic RNA is translated into the open reading frame 2 and 3 proteins. The open reading frame 2 protein combines with the genomic RNA to produce new virus particles, which egress from the cell, which leave the cell and undergo maturation. So this really completes the life cycle, and the new virus is then ready to bind to fresh cells. There is some evidence that the ORF3 protein may associate with intracellular lipid membranes to cover the virus particle. And then once the virus leaves the cell, the maturation process is for this lipid membrane to disappear, exposing the capsid, which is made mainly of the ORF2 protein. We utilized the fact that the virus, if you express open reading frame 2 protein, truncated versions of it, you can produce virus-like particles which resemble the virus in its structure, especially in the exposure of the region shown here in blue, which is the region that binds to the cells and binds to the receptors for the virus to enter. This is also the region that is target of neutralizing antibodies to the virus. So we produce these virus-like particles in E. coli, in bacterial cells, purified these virus-like particles and used them as tools to study the HEV receptor and the entry pathways. I'm omitting a lot of the work that we did with the open reading frame 2 and 3 proteins. All that is published and part of HEV literature now. So these virus-like particles, we showed that they bind to both liver and non-liver cells, but they bind much more efficiently to liver cells than to non-liver cells, as shown in the binding curves. When we take these particles and bind to the surface of cells under conditions where receptor-mediated endocytosis is inhibited, for example, if you incubate the mixture at cold temperatures, you can see that the virus primarily binds to the surface of the cell. But when you move the cells to 37 degrees centigrade, very quickly the virus enters cells and can be seen as particles within cells, as opposed to particles on the surface of cells when you keep it at cold temperatures. So this told us that the virus was binding and it was entering cells. So to identify the receptor, we again carried out binding kinetics on 3 different types of cells: HUH7 cells, A5, which is a liver cell, A549, which is a lung epithelium cell, which had been shown by others to support the replication of HEV, and on HEK293 cells. And as you can see, both HUH7 and A549 cells bind much more efficiently to these virus-like particles than HEK293 cells. And just to cut a long story short, I'm showing you, I'm showing you the final experiment where we took biotinylated cell membranes incubated with virus-like particles, and by pull-down assays and followed by mass spectrometry, found that there were 2 different proteins that were specifically coming down with these VLPs. One was a 69 kilodalton protein that was later identified as riboforin, and the other was a 56 kilodalton protein, which was identified as the beta subunit of ATP synthase, which is also present in the cell surface. And through many other experiments that involved both siRNAs as well as antibody blocking experiments, we showed that ATP5B acts as a receptor for the entry of HEV. And again, this is published. Now, the question we also asked is, how does the virus enter cells? And this is a cartoon that shows how most viruses enter cells through either phagocytosis or micropinocytosis, or various endocytic pathways that can either be clathrin dependent or caveolin dependent, or clathrin and caveolin independent. And again, we did a number of different experiments which are summarized in this cartoon here. What this shows is that once the virus binds to the cell surface receptor, there is aggregation, and the virus gets into endocytic pits, which are lined by clathrin molecules. Once that happens, actin and dynamin come in to pinch off the endosome, and the virus goes into an early endosome. It is this early endosome that then allows the virus to uncoat, possibly based on pH changes in the endosome, and subsequently the HEV RNA is released. So the early events showed very clearly that it's an endocytic process, and it's a process that depends on both actin and dynamin, and also another kinase which is called phosphatidylinositol 3 kinase, or PI3 kinase. Some of the later studies that we did with hepatitis C patients were based on the understanding that the host response possibly determines the outcome of disease. In most people, HEV is a self-limiting disease, but in a small fraction of people, it becomes fulminant. So we reasoned that it is likely that these differences in the host response can be seen in the on proteomic and metabolomic analysis of both self-limited and fulminant hepatitis patients. And if identified, we should be able to use this for clinical value. The approach we used was to look at the proteome and metabolome, both in plasma and urine, using 2D gels, eye track, and mass spectrometry. And metabolomics was done mainly with nuclear magnetic resonance studies. And again, I'm summarizing a lot of that data on this slide. What we found was that compared to self-limited hepatitis, in fulminant hepatitis, there were essentially 4 classes of proteins that were differentially regulated. Complement proteins, proteins involved in lipoprotein metabolism, a group of protein called lipocalins, and acute phase response proteins, both negative as well as positive APPs. On this slide, all the proteins that are shown in green are upregulated in fulminant hepatitis, whereas all the proteins that are shown in red are downregulated in fulminant hepatitis. So these were the really the outcomes of our proteomic and peptidomic studies. The summary of metabolomic studies are shown on this complicated chart, which essentially shows us through main cycles, metabolic cycles, that are modulated during fulminant hepatitis. One is the TCA cycle, and the other is the urea cycle. And another pathway that is modulated is the branch chain amino acid synthesis pathway. We had also characterized earlier effects of hepatitis E virus on glycolysis by understanding how the viral proteins change the levels of glycolytic enzymes, both phosphofructokinase as well as lactate dehydrogenase. Our metabolomic studies characterized several metabolites. The ones shown in green are downregulated, the ones shown in red are upregulated. And you will notice that acetone, which is involved both in ketoacidosis as well as has been found to be, and ammonia, which has been found to be involved in the effects of hepatic anomalies on the brain, were found to be differentially regulated in some of these studies. So based on many of these studies, we also asked about this elusive question of the role of hepatitis E virus during pregnancy. As you know, the virus causes excess mortality, especially during the 3rd trimester of pregnancy, and this has remained an enigma. What we found was that if you look at factors that are related to pregnancy, there are several things that happen. There is increased estrogen, there is increased progesterone, there is a shift from Th1 to Th2 response, there are changes in amino acid levels, folic acid deficiency, serotonin decrease, as well as increased HCG. And when we looked at factors related to hepatitis E from our proteomic metabolomic studies, we found that there was increased estrogen, there was increased progesterone. There was also a shift from Th1 to Th2 and changes in amino acid levels, especially in branch chain amino acids. So the hypothesis that we built was that hepatitis E just exacerbates pregnancy by acting on some of the same pathways, and therefore causes additional imbalances during pregnancy, which leads to a worse outcome. So let me come back to summarize what I have told you. And these are the summary of our molecular studies. We showed that heparin sulfate proteoglycans act as attachment factors for hepatitis E virus to bind to cells. We also characterize ATP synthase beta subunit as a potential HEV receptor. We did a number of studies to look at how the virus enters cells and characterized dynamin, clathrin, and PI3 kinase dependent endocytic pathway through which the virus enters cells. We carried out many studies that I have not told you today, but that's all published on the effects of the open reading frame 3 protein. We expressed the protein, characterized it, found it to be a phosphoprotein, showed that it modulates the MAP kinase pathway and affects growth factor signaling. In summary, ORF3 protein promotes cell survival. And the idea is that if an infected cell lives longer, it produces more virus particles. We also carried out studies on open reading frame 2 protein expression, analyzed it and showed it to be a glycoprotein, and produced virus-like particles using this protein, which allowed us to map the receptor and early endocytic pathways. So let me finish here by again thanking both the Russian Academy as well as CDC for selecting me for as one of the winners of the Balаяna medal for this year. I'm truly honored and deeply grateful. Thank you very much for your kind attention.
Special thanks to Dr. Jamil for his interesting report about molecular study of virus of hepatitis E. It's always very important to listen to the information from the people who developed this, one of the authors of the information and the author of the study of hepatitis E, and we are really thankful for that you introduced this very interesting report. And because your report was in a video script, if there will be any questions, we definitely contact you and we ask you to answer them. And now I would like to introduce the next nominee for the medal named after Mikhail Balayana. This is our friend Yuri Karyatni, and he is given the award for his work in the study of virus of hepatitis E. And I should say that Yuri was working in the group of Mikhail Balayana directly with him, and many of results which were received with his participation. And I'm really happy to give this diploma, this medal, which you are awarded. You will receive them via post. We send it later. So Yuri now is living and working in the United States of America, and his time zone is, I think, 3 at night, and we are thankful that he is online, and we hope that after his report, if the listeners would have any questions, definitely will be some questions, you will be able to answer these questions online. So Yuri, please, the report about the infection of hepatitis C as zoonosis, the history of opening the perspectives of the study. So good morning, good day. I would like to thank, for me that's a great honor to introduce my report here, and I would like to thank the organizers of the symposium of the virus hepatitis and Mikhail personally, with whom we have 40 years of cooperation and friendship. My report is mainly dedicated to, it's quite more historic. The point is that my virusology study and career were so developing that I studied a lot of viruses, and the virus of hepatitis E was following me all my career. more than 30 years, and I worked over other infections, and suddenly hepatitis E virus appeared, and I think that this is the most difficult, the most riddle infection. That's why my subtitle of my presentation is Virus Hepatitis E Infection, Enigma, Questions, Answers, and another question. So I started to work over virus hepatitis E when I was invited as the senior scientific specialist in a group of Balаяna, and I was the head of serological group. And this is quite an old slide. But when I started to work over this problem, the classification of the infection of viral hepatitis was presented on this slide, and that was based on the classification not of a virus, but more on the common sense, on the clinicals, on the experience, and how the doctors described that. That's why mainly that was split in two main groups in the way of transfer of infection. This was enteric or parenteral with the blood, and the group of hepatitis with enteric way of transfer were the hepatitis A and hepatitis E. At the same time, when I started my work in the lab of Mikhail Balayan in the institution of poliomyelitis, my functions were mainly to work with hepatitis A. That was the creation of population immunity and serological support. At that time, there were vaccine developed, the new diagnostics for hepatitis A. There were a lot of field studies and work, and with hepatitis E, Alexander Angeparidze was working, and he is also going to make report. He is my colleague, actually, and big friend. And I was a little bit envy, because hepatitis A was was found many years ago, and with hepatitis E, Mikhail Balayan was opened recently, and that was very interesting, very attractive to work over this problem. So when I came to the department, that was a fantastic organization. On this slide we have the achievements of the department, which consisted of 40 people. More than they have, they were candidates or PhD, actually, the doctors. There were 4 laboratories inside. And if we study this list, it would be difficult to find the organization which, within 20 years, starting from the ground zero, with before Mikhail Balayan, no one was working with hepatitis in the poliomyelitis institution. So the main scientific achievement and the work was difficult to overestimate. And the first isolation of hepatitis A virus, and after that the development, the modeling of the first model, Lab 1, on the non-human primates, the first pig model, and also the first works on the definition of the zoonosis nature of this infection. Considering the hepatitis A, there were also a lot of achievements, and this is the isolation of hepatitis A virus from non-human primates, and the same as with hepatitis E, and these were the studies inside the vivarium and modeling among primates of the HIV outbreak. The first production of IgM diagnostic kit. Then also that was the first time in the Soviet Union, the first national surveillance program for development of the population immunity for the HIV. That was for the whole Soviet Union program. That was for the collective immunity, and the first HIV national inactivated vaccine. And I had to work 2 years working only with hepatitis A, but with the received data, they told us that at that time hepatitis E was called as hepatitis A2, and that was quite similar, but at the same time, while studying epidemiological parts, there were found different fundamental differences, and at that time the global distribution was split in 2 groups. These were the outbreaks with the clinically expressed picture. The main outbreaks were clearly defined, and also because there were developed some primitive, some simple, based on immune and electronic microscopy serological tests. They were started to found the serological of, in some areas, epidemiological evidence were no outbreaks. That was one of the most important questions, and Dr. Meng in his report focused on this topic as well. But the clinical courses, the clinical situation and the immune response, as you see on this slide of the hepatitis A, they were quite similar. But that was interesting to work with Mikhail Balayan, and I have never seen the scientist who knew all the literature. Just he kept all this in his mind, and in order to work on equally, to have the discussions with him and disputes, you must have followed all the scientific literature on the topic. That's why I started to focus on what is different in these 2 infections. There were long several years of such discussion with Mikhail Balayan, because he normally disagreed at the first glance to some suggestions or ideas or hypotheses. That was a very big challenge. And when we started to compare the hepatitis infections of A and E, we found out that the clinics is very, very similar. But hepatitis E at that time mainly was introduced by the water outbreaks in the developing countries with a hot climate and with low sanitary rules. One of the peculiarities which still we cannot respond fully and correct, this is high mortality among the pregnant women. And if we compare the outbreaks of hepatitis A, then this is explosive in the volume of disease cases, then it goes down, and after that It's like a tail which continues up in some outbreaks up to 1 year, and this is based on the transfer of the infection from human to human by the contact way. With hepatitis E, that was not found out. The outbreaks were continued in different volume from 2-3 months up to half a year, and then there was not documented data about that the infection can be transferred from one human to another one. And also, quite often, we couldn't find the connection between these outbreaks. These outbreaks could be located at a small distance, especially that was in the Middle Asia, but they were like as spots. There were no reservoir of human beings. And then there was a question that can be zoonose, and the virus between these outbreaks. This is not a human infection, and in order to make the infection expressed, the dose of the infection should be high. So it means that there should be some reservoir which would give this virus contaminating mass, contaminate the water, and after that there can be the outbreak. Next, one of the answers, the questions, was where we have antibodies in totally different areas in the Middle East, in Europe, where we don't see these outbreaks. And this discussion with Mikhail Balаяna that was lasting in 87, within half a year. Within half a year we had discussions, does it make sense to invest the efforts into the zoonosis study of hepatitis A, our meetings, our money, our resources, and as a result we started to make historical studies of what used to be before, and we know that hepatitis, enteral hepatitis, is always presented while the war activities, and I made the historical literature analysis and I introduced this in the university. The first point of historical feedback, that was the March 19 of the March 1799. That was the Napoleon Army, and that was his African campaign, which actually finished in the Accra city. This is in the northern part of the modern Israel, and his army was attacked by the virus hepatitis, and that's difficult to imagine that in the end of the 18th century, France, and this is the adult population, and there were not any immunic people presented to hepatitis A. We know, and we studied, and we had the data on the Southern Europe, on the presence of the immunic structure to hepatitis A, and normally up to 20-25 years old, most than the half of population have the antibodies. It means that's probably or highly likely that was the outbreak of the virus hepatitis E. The second fact, that during the North African campaign in the Second World War, German and Italian troops suffered from described cases of enterically transmitted hepatitis, and that was especially in Italian army. And if we take the history of the Second World War, it is well described, especially Italian army was suffering heavily because the sanitary conditions and the conditions of the troops and the march, and the situation in the army was quite low. I mean, the situation was really low in comparison to German army, and the people who were presented, and that's difficult to imagine that they didn't have the immunity against the hepatitis A. That's why it was supposed that was mainly hepatitis E. And also Mikhail Balayan had a lot of materials because he was talking to the militaries during the presence of the Soviet military contingent in Afghanistan. He was involved in the work there, and he shared the facts. And I have seen these soldiers who had two hepatitis, hepatitis A, and then after that there was hepatitis E. What was interesting is that mainly they were suffering the parts of the army which were in the mountains and who drank the water from small springs and small sources of water. Studying all this data, we came to conclusion that mainly, and from small whales, we came to conclusion that if this is a zoonosis, then that should be quite big amount of these animals who make this reservoir, and highly likely these are the rats. And in the group of gepatitis there was developed already the pig model, swine model, but in the Middle Asia, in the Central Asia, in Afghanistan, the swine model, for some reasons, that could not play a role as a reservoir because of religious and ethnic peculiarities and reasons. The pigs, they don't have the pigs in the Central Asia, and after long debates it was taken a decision that we should find isolated outbreak where could be only one source of water, and to go to make the hunt for these animals, I mean for the gnowers. And Mikhail organized with this with ministry because we needed the mass of different moments to organize, and I flew on a plane to Kyrgyzstan, to Kyrgyzia. That was first Bishkek, and Dr. Usmanov, our big friend and colleague, who was working at that time in Bishkek, he organized the expedition to Osh region, where at the time was observation of massive outbreaks of hepatitis. That was not hepatitis A, that was highly likely hepatitis E. We arrived to Osh city, and then with to city Osh, and with the anti-pest support we have organized the expedition, and they were thoroughly chosen the area. This is an ancient area. This is Jigdeli. This is hundreds of kilometers from Osh. We had this truck. We were driving the whole day. And this is not Jigdeli, but this is rural area of Kyrgyzstan, quite similar villages closer to the border with China. And that's how it all started. That was November of 1989 in this Jigdeli village in the Osh region of Kyrgyzstan, with a population of 625 people, 39 people within a month were found acute virus hepatitis, and one pregnant woman died. And I witnessed that. This is an awful picture. Within 24 hours, absolutely normal, absolutely healthy looking woman. She became yellowish skin and she died. We started to hunt the different samples of wild rodent near the. There was only one water source, one spring from the mountains, and within 2 weeks we have collected almost 500 rodents, and then I found out that these gnawing rodents, they do not take water from the zoologists, and the whole water is out of their food. That's why blood is very thick, and out of these 500 rodents we managed to get at least some blood or serum out of 23 samples. When we arrived, these 23 samples were controlled by electronic immune, and in 5 of them there were antibodies found. The next stage was the development of the model. They were taken the lab rats, which also by the method of immune electronic microscopy were controlled on the presence of the antibodies for HIV, and they were all negative. We also used two isolates. One isolate was from the patient, and the isolates from Mikhail Surenovich, Balаяна, and one from the monkey, from macaque, and this was a model which was developed by Dr. Japaridze, and you see that this, on the 4th day, these animals started to excrete the virus, and that continued up to the 23rd day. And then it was developed, the model of the lab rats, which were increting the virus, and there was the parallel test on the seroconversion, and all the rats have produced antibodies for HIV. They became immunopositive. What we did not notice in this group of animals, which were infected by the extract of the healthy person, the next attempts to pass this virus were not lucky. That was also not the similar result was received on the monkeys, not similar to humans. They could be maximum 3 passages, and that can be called as the negative attenuation of the virus. And because the virus has the zoonosis nature, it's not a human, and to pass into the human population it cannot, or it happens very rarely. This is how it looks on the immune electron microscopy. This is hepatitis E viral particles, and there are also antibodies which are taken from the seropositive sample from the patient. In this table, we sum up all the first experiments of different groups for the modeling by the method of the definition of the zoonosis nature of the HEV. The first swine model was developed in our department. That was managed by Dr. Balаяna and Dr. Japaridze. Then it was repeated in another group of Dr. Karan in America, Corcoran in America. Then Dr. Usmanov repeated that experiment in Bishkek. But I would like to say that more than one passage, unfortunately, they couldn't produce. The only striking thing is that one of the swines which was infected, it has shown that was quite a heavy hepatitis E, but that was only one case. And the first information about this zoonosis that were the results of this expedition to Kyrgyzia, and then they were continued to work with the swine model that was repeated by American specialists by Army Institute in Yemen, and there were also several experiments by Dr. Usmanov 6 years later, successfully repeated this experiment. At that moment, the virus was fully sequenced, and that was developed the strategy of the virus genome. And my participation actually was over in Russia, and I moved to Israel, where I was the head of group of the virus hepatitis in the Central Virus Laboratory of the Ministry of the Healthcare in Israel. And generally, at that time, the government of Israel was highly interested in virus hepatitis for two reasons. The first one, the big migration from the former Soviet Union countries. Within 2 years, more than 1 million of people arrived, and their immune status, was not clear. That's why there was arranged the group, and in order to study these people on the presence of the immunity to different infections. And the part of the hepatitis A, that was the part of the project which I was given to make. The second reason why government of Israel was so interested in hepatitis A, because the Jews and Arabians live close to each other, in Jews part is well studied. The Arabian part, there is always the presence of different diseases. Quite often they refuse from the vaccination to the poliomyelitis, and this is well known that virus of hepatitis A and poliomyelitis, these are viruses, Bicorn viruses. They are connected, and that's why we invested a huge amount of resources into this program, and that was quite easy to gather the samples out of the donors, or soldiers, or the population, because that was the state program. And after we made this work, I should say that we thought, why not to study what happens in Israel with hepatitis E. And I found two local publications that in the 50s there was an outbreak of unknown hepatitis in Jerusalem among the Orthodox group of population, where several pregnant women died. And in the middle of the 70s, In Israel, there was a similar outbreak of hepatitis among the army, and that was connected to the water pipe accident. And at that time, my group actually studied the whole immune structure of population in Israel, considering the hepatitis A, and 50% of population at the age of army age, from 18 up to 25 years old, they have the natural antibodies against, received from nature, against the hepatitis A. And that was difficult to suppose that these cases in 70s, there were no diagnostics, but that was stated as hepatitis A. But we have established our former connections in Soviet republics. Dr. Hodikov, Dr. Favorov, who made report, also at that time they used to work in CDC, and Dr. Fitz also CDC gave us the reagents for the ELISA analysis for the antibodies for HEV IgG class with approval and IgM. At that time, that was the best test which you could get, very specific, because other tests like Genilat, for example, we also tried to use it. It gave too many non-specific reactions. And in this very table, there is a sum up of the immune structure of the population in Israel. And it found out that among the Bedouins, the people who are very close in connection to different types of cattle, they live in desert under tents, and they are surrounded by the millions of different wild rats, and as you see in the table, the immune structure, they are surrounded by rodents, and you see that the percentage is much lower in all the age populations and groups. And in 2015, in 2020, I visited the central lab in Israel, and they have expressed the virus of hepatitis E in Bedouin village from the camel. That's why that was not direct approval that the groups of the population who touch or have contact with the zoonosis reservoirs, they, in their percentage expression, the antibodies are on a quite high level. At that time, it was approved by the group of Dr. Purcell in National Institute of Healthcare. They made the inspection of Bedouins, not in Israel but in Egypt, and they, by other method, they used the other method, but they received the similar results. On this, the part of my involvement in hepatitis E in Israel was over. I moved to United States, and I had the apprenticeship, and I was working in parvovirus laboratory of the University of the Iowa State. That was the diagnostic lab, which also was a reference lab for the Middle West of the United States, but for not clear parvovirus B19 endemically in children organizations and children gardens in the kindergartens of the Middle West of USA, and the main task of this study was there were sick adults with dominant hepatitis were found in the liver of these people were B19 found, and I was working with B19, and I didn't have even any thought that hepatitis E can be found in the center of United States, in Iowa and Illinois states. And then there was a historical event happened. Almost a revolution. Dr. Meng, working in VIH, expressed the virus of hepatitis E from the swines. And I should say that for the Middle East states, especially Iowa, or Illinois and Nebraska, the production of the swine, this is industry number 2 after production of the corn, maize. The whole population in the agricultural areas in the states, they actually are busy in this sphere. That's why the expression of the new virus, which can potentially be dangerous for people, caused very wide public discussions, because that could be the direct influence. People were worried because the swine could be a reservoir of the infection, which is dangerous for people. And here in this table, I have a sum up of the data after this discovery of Dr. Meng. Once again, this is a revolution discovery, because that was the first person who expressed virus and kept it in the hand, because before all our hypotheses, they were approved only serologically or on the models, but that was obvious that hepatitis E, it's not a human infection, this is zoonosis. And here we show actually natural reservoirs and the lab models, and you see here that there are rats and swines, and this happens from different groups in Asia, in the United States. Dr. Favorov defined the seropositive rats in Baltimore. This is 40 kilometers from Washington, capital of the United States. So from the whole world, we started to receive more and more data about that the virus can circulate in different animals. And that we must have used. Being in the center of the country, we found out that hepatitis E is endemic for United States. This swine infection, and there is a constant outbreak in these farms, because as Dr. Meng told, that the constant population of these farms, these sows who have the process of reproduction, this is insemination, artificial, they get infected, and in 6 months these swines, they are killed, they are slaughtered, but the sows, they are constantly immune, and the virus is constantly circulating because of them. And that's why we cooperated once again with our old connections with CDC, with a group of Dr. Fils and Gordichuk. They provided us reagents. That was the same test based on mosaic protein to hepatitis E. and there was a new version, more sensitive, more specific. We also cooperated to our clinical specialists from University in Iowa and Central Clinic Lab, which made very positive influence on the way of this work. We received 3 grants from the government of Iowa, and that led us quite fast to start these studies. From the clinics, we had 204 patients with different of hepatitis non-A, non-B, and there were toxic hepatitis, included different diseases of liver of not clear nature, and first there was a screening done for the presence of IgG, and almost 5% of patients had the antibodies at the same time. We're sorry to interrupt you, but we have the reglement, we have the time frame. Yes, and I finished. Then further they were studied and inspected using the PCR, the swines, and there were similar results with Dr. Meng, and the young pigs, the age of 3 months, they gave the seropositive results. So that was a proven the permanent outbreak on the farms. Then we sequenced the results, and they were very similar. That was the virus of the 3rd type, and they were similar to S1. This is from the first patient which was expressed in Minnesota in the clinics, and then there is the genealogical tree with the virus of 1st and 2nd type. With this grant, we studied the sewers from the farms, and you can see that at the exit there was found the virus of hepatitis E. That was also approved. The virus was expressed from Ivor River and from Agaya River. This is the map. This is the same slide, but there is expression of the viruses in Europe, in Israel, in Asia, and of course in China, in India, and even in Australia and New Zealand and United States. And this is the modern phylogenetic tree, an idea about where there was the virus expressed, and now there is the family of consisting of 2 subgroups, and we have there is the whole group of the viruses of hepatitis E. On the next slide, it is shown of the areas or how the virus can circulate, how it is received by the wild animals, by the domestic animals, and how it is transmission to human beings. And I think that I finished with the viruses of hepatitis E, but working in the organizations which make the outsourcing, helping, and assisting the clinic studies, and there is such a definition of the virus safety panel. There are defined the infected from the viruses, and there is a list of them, and every year there are added new and new viruses. And in 2014 there was added hepatitis E. That was my final comeback to the work with virus hepatitis E. And in conclusion, the virus of hepatitis E can cause infections, asymptomatic infection and disease, and also in different groups of population, even in not endemic areas. And of course, the group of the risk have more high level of antibodies and transmission and antibodies prevalence, and that can also include the group of population who, for their duty or the work, they have a contact with the domestic or wild animals. And of course, hepatitis E, this is a zoonotic infection, and the significance of the immune structure of the population gives us big data about the distribution of this infection. And I think, and there is the group of the specialists, of the researchers, of our core workers. This is Institute of Polymyelitis, this is Central Lab of Israel, the University of Iowa, and State Hygienic Laboratory of the State of Iowa, and CDC. And I think that I won't come back to hepatitis A. And in 2017, these are my children, my daughter, who used to be a student of the university in Virginia, where Professor Meng is working, and she had the training and apprenticeship in another lab of Dr. Allen, and for me that was surprising. And surprisingly, in one of the publications, I find my my last name, but not my initial. So that's a small gift from my daughter, hepatitis E. It's transferred in a generation and work. Thank you very much. Ready to answer questions. Thank you very much, but we don't have time to answer the questions. And I thank you, dear Yuri, for your support, for your work. We know your biography, we know your work, and that was a proper decision of the committee to give you diploma and the medal. We will send you in the nearest future. Thank you very much. And it was a great pleasure. I would like to introduce and to turn the floor to the next person who is awarded. This is a new generation. This is the Dr. Ilya Gordichuk, a member of the Institute of Polymyelitis. He is of our group member, and he is given the medal of Mr. Balаяna for the study which he is making right now on the hepatitis E, in particular on making the vaccine. And one of the most important things is the preserve of the Mikhail Surenovich Balаяna work. This is the colony of monkeys. With Dr. Polishuk, he made this colony, and which survived through the most difficult times in our country, and I hope that these times are over. We used to survive, and we managed to save this colony, and right now Ilya is actually he continues the work of Mikhail Balаяna. Hello, I'm really thankful for Mikhail, and I'm thankful for the committee for such a high award. And my report today is mainly the next chapter of the Yuri Karyatni, because in the lab of Mikhail Balаяna we continue to work over the models. We love this slide because it shows that in the scientific life of Mikhail Balаяna there was a difficult moment. I hope that you can see when on the stage of the 29-40 days he was almost in despair to express virus of hepatitis E from himself, and he stopped taking the samples, and there is a small gap on the graphic, and when he felt sick, he continued to take the sample that was described hepatitis E through this, and many of you know that Mikhail Balаяna and was the founder of this, but not many people talk about his rheological work. And Mikhail was also making the infection of monkeys, and he found the first colony of the monkeys. That was the first colony of them on territory of Russia, and we actually continue his heredity. And I'm going to talk today less about hepatitis E virus. I'm going to talk about the models which we are using. This is the colonies of the monkeys, which are marmosets monkeys. And since the moment of the foundation, this is the maximum amount of the colony, and still we work with them, but the modeling of the virus hepatitis E with the infection, it is a very serious task, because as you know from the previous reports, the genotypes of the virus are differently behaving, and the most distributed genotype of the virus for the human beings is 4th, but autochthonous cases in Europe, this is the 3rd genotype or the 4th genotype. These genotypes are transferred like zoonoses, but the lab model is very serious problem. For the virus hepatitis E, we need a model which can help us to make the studies, to model the hepatitis itself, the infection of the liver. We need a model which is well characterized from the rheological point of view, and the response for the vaccine, and the response for the humoral response also, so that we could estimate the rheological and physical changes. And the marmosets are quite convenient in this case. On this slide we have detailed description of all the pros and cons of this model, but some of the minuses, some of the problems we managed to overcome in due course. And briefly, one of the most important factors which make the marmosets the ideal, almost ideal model for modeling the many infections is that they quite often produce the twins, 2 or 3 babies, and we can make experiments for the control group which will consist of twins. And I give you such an experiment as an example today. And moreover, for a long time we are working with the T-cell response on the marmosets, and this we have results in the article where we studied the choice of the clones of the antibodies for the coloring of the markers in the marmosets. And I can give one of the examples how at the present time we characterize the T-cell response. We characterize it in experiments which are done with the vaccines against the virus hepatitis E. At the present moment, we unfortunately do not produce the differential diagnostic of the response, but low proliferation on the antigen stimulation is quite well described on this slide. We express the T cells for the marmosets, split in C408, and these are non-stimulated cells. They are in rest, in the proliferation. Then in the control samples, you see the populations divided, split, and at the vaccine animals with the stimulation of the virus proteins, we see significant proliferation of them. As far as high reproductivity of results of biochemical study of blood of the marmosets, I also introduced the results of the studies of the state of safety of our antivirus vaccines in our lab, and we see that similarity of results, and these are black dots in the control group, 12 animals, control group 6 animals. The reproductivity of the results is really high in the results which we get from the small lab animals. On RT on Enigma, and it is shown also the reference parameters of this result, the same results you see on the epidemiological studies and the amount of erythrocytes. And I have taken one most frequently used methods in our lab. So I will give you an example of how the marmosets are used for the modeling of the virus hepatitis caused by the third and the first genotypes which are in the immunic outbreaks are mostly noticeable. I will not talk about the theory of hepatitis E. You all know this quite well, and you know quite a lot of this, really. So we used as many other developers of the vaccines were developed on the basis of this green color CP. That was work started by the students of Mikhail, by his group, and as the initial work of the infection of marmosets, the same actually. People were working on this and how we used the full, not full protein, or RF2, we used the fragments from 110 up to 616 on the slide. These are the fragments which were used in other vaccines. In heparin, the vaccine registered already. This is P239 vaccine, which was registered and approved. The synthetic protein, which was received with the usage of this gene P or F2, that was for the production E. coli, and that was substituted for the part of the protein. So, and I skip all our stages of cleaning, of preparation of the protein. We managed to get the samples, to get the particles, which were approved by the method of electronic microscopy. This small, nice globulus, or the balls, which are different to the native virus of hepatitis E, which Yuri has shown us in the previous reports. This is virus-similar particles which consist fully of this protein RFO2, and this is quite stable. We controlled how it is in the storage, and after that we made the experiment immunization of our marmosets. We infected them after the first genotype of virus of hepatitis E and the third genotype expressed from the swine, the sow mother. The animals were infected in veins, and every day after that we expressed the RNA of the virus of hepatitis and also antibodies, Ig for the virus, E, and biochemical parameters. In this study we used 6 animals. We knew how we could reproduce the virus hepatitis E in monkeys, but we didn't know definitely. That's why we used both genotypes. And on this slide it is introduced the results of particular level of the antibodies of the animals, and I should say here that the level of the antibodies in this situation has an expressed title, because as we know from previous reports, the development of the model for any infection is a long way, and because we cannot use the international standards for immunization on the level of the antibodies, because this standard is the human serum with human antibodies, and unfortunately this is quite specific for the marmosets, only for this species. That's why we need to use the new reagents and to develop them. So the animals which were not vaccinated, they are in blue color, and before the virus challenge there didn't appear antibodies specific for hepatitis C, while the vaccinated animals, you see the significant increase of the level of antibodies. The interesting moment is that if the virus infection, we caught this point where the growth of the level of antibodies happened to in the control group of animals after infection. Also on this slide it is seen that after 151 week after vaccination and the challenge, the animals' level of antibodies stayed the same, 2.5. What is even more interesting is that the animals which were not vaccinated but were experimentally infected with the first and third genotype, it's typical for the human infection and also for animals infected with the first genotype expressed RNA in phagus from the 2nd to 6th week, and the animals which were infected with the third genotype, they expressed the virus RNA in fecum from the 2nd up to the 3rd week. It was a shorter period of time, but not any vaccinated animal did not express in fecum the virus in fecus. And we see also here the immunized animals with the black dots, and the animals in placebo group. We see that the animals received placebo at the first and the third genotype. There was a transferase increase, AST, LTG, but at the same time we don't see the significant growth of general bilirubin in these animals. So based on this data, we suppose that some average level of destruction of animals in liver actually occurred. generally, that's not such a big volume of data, not a huge experience of work with hepatitis C as on previous report makers. So I feel really modest in this company, but we continue, of course, the work of Mikhail Balayan, and in conclusion, I should say that the marmosets were really infected by the genotype 1 and 3, and this is very practical model in the reproductivity of the vaccine. The West is skeptical. What is the positive thing in the model in this list? These are my co-authors and colleagues, and of course Mikhail Balayan is constantly with us invisibly, and thanks to him for this discovery of hepatitis E, and he actually created the colony of the marmosets, which is a result. With a long period of time it turned out that he was right, and this is the optimal model for the work with this virus. Thank you very much. Thank you very much, Ilya, and the diploma and the medal is waiting for you, and I hope that in the nearest future, in a couple of days, we'll see you. You will visit our lab, and we will give you in the honorable situation. Of course, the colony of the marmosets is crucially important, and we should remember not only Dr. Balayana but Dr. Polishuk, who saved this colony, and many thanks to you. You're a representative of the new generation in hepatitis E study, and I'm really happy that we are working together, and we hope that we will continue our work. Many thanks for your interest in the report, and now, because we are out of our time frame, we, without any break, we move to the next session of our meeting, and all the 4 report makers which will be, they all are the winners of the medal of Mikhail Balayan.